The aim of this procedure is to measure the reverse transport of dopamine in a neuron that is voltage clamped, using simultaneous patch clamp and amperometry to measure DAT-mediated DA release. This is accomplished by first testing the integrity of the amperometric electrode with dopamine. The second step is to place the amperometric electrode near the cell and achieve a whole cell patch of the neuron with a patch pipette. Next, the voltage of the cell is controlled with the patch pipette, and the DAT-mediated dopamine release by the cell is recorded with an amperometric electrode simultaneously. The final step is to add the DAT blocker and record the whole cell and amperometric currents again to allow the currents specifically mediated by the dopamine transporter to be subtracted out.
The significance of this technique is the ability to measure reverse transport of dopamine via the dopamine transporter while simultaneously controlling the voltage of the dopaminergic neuron.
The simultaneous patch clamp amperometry recording system requires an inverted microscope with excellent DIC optics and a long working distance lens. To generate a DC light source for the system with low electrical noise, connect the microscope lighthouse to a car battery. The hydraulic micro-manipulators are used because they do not appreciably contribute to the noise levels. In our configuration, a right-handed manipulator is used for the voltage clamp whole cell recording and the left-handed manipulator for the amperometer.